Okay, lecture 8 will talk about the structure of proteins, what they are made out of, and how their structure changes when they try to maintain their structure and the function. So hopefully this will be a review for most of you guys because you learned this stuff through your previous biology class. First of all, proteins are made out of amino acids, right? And there are 20 different amino acids, you remember that. And this is a typical structure of an amino acid. So there's an alpha carbon in the middle of it, the center, and then there's an amino group, and then carboxy group, and hydrogen ion there. And then there's a side chain, R. This side chain kind of differs among 20 different amino acids. So what's different among 20 amino acids is based on what you have here, okay? So then those 20 different amino acids are right here. Okay. So the properties of R, the side chain, determines the structure and function, interactabilities of these amino acids, as you know, right? So 20 different amino acids, but you can divide them into 4 different groups again. Okay, so neutral or non-polar amino acid are these guys: glycine, alanine, valine, isoleucine, tryptophan, phenylalanine, proline, methionine, and leucine. So these are non-polar or hydrophobic amino acids, hydrophobic, non-polar. So if you look at the side chain, there's no polarity. So they are strongly hydrophobic, so they don't interact with water. So when these amino acids are added to protein, they will try to stay away from water. So normally they are found from inside of the protein. And then there are acidic and basic amino acid. So these are charged amino acid. Okay, so acidic, aspartic acid and glutamic acid. So their name says they are acids, right? So they can donate protons to the environment under the neutral pH. I mean the physiological pH, CO minus. They have the common structure. So they are kind of negatively charged on the physiological pH condition. And then these are the basic amino acid. So lysine, arginine, and histidine are 3 basic amino acid. If you look at their structure, okay, they have positive charges there. Okay, NH3 plus, NH3 plus. This one has the positive charge as well. So these are kind of positive charged basic amino acid. So they tend to receive hydrogen ions under the physiological pH. That's what they do. And then there are neutral polar amino acid. Polar meaning what? They have a polar electronegativity positive structure, so they will interact with water molecule. So these are strongly hydrophilic amino acid. They can form hydrogen bonds with water molecules. So these are serine, threonine, tyrosine, asparagine, glutamine, and cysteine. These are all hydrophilic amino acids. So you probably need to remember and memorize these amino acids since you are in the supplement level class. So you need to know all 20 amino acids and which one belongs to which group. Okay, and I want you to memorize their names in each group and hydrophobic versus hydrophilic, acidic, basic. And in your first exam, we'll have a question about this. You will be asked to draw at least 1, 2, 3, 4, 5, 6, 7, 8, maybe 8 amino acid structure at least of your choice, and you will be asked to link them through peptide bonds. You'll be asked to label them, their names, and the alpha carbon amino group, carboxy group, and the side chain structure, and all those things. Okay. So when those amino acids are linked together, they are linked together through a strong covalent bond. We call them peptide bonds, right? So there's one amino acid, there's the second amino acid. Between those amino acids, you see a carboxy group and amino group of these 2 amino acids coming together. So the water is removed between them, and you see the linkage between carboxy group that has only oxygen left there, amino group that has, you know, the hydrogen left there. So that's the peptide bond we're talking about, strong covalent bond. Made through the sharing of electrons. Okay, so the peptide bond is the key covalent bond in forming protein structure. They can also form disulfide bonds between 2 cysteine residues. Okay, so Cysteine, remember the structure right here. Cysteine, it has SH side chain, right? So this cysteine can form a disulfide bond with another cysteine when they are in close contact with each other. Okay, understand that. So methionine has S, but there's no SH there. So there's a difference between methionine and cysteine. So that's why methionine cannot form disulfide bond, but cysteine can. Understand? So if you have cysteine here and another cysteine there between SH, when they are oxidized, the hydrogens are taken away, and you form disulfide bonds. Okay, so oxidation will form disulfide bonds, but if you reduce them, the disulfide bond could be broken up. Okay, so the oxidation of two sulfur hydride groups form disulfide bond, just like that, which is also a covalent bond, which is a strong bond. Okay, but if you provide a lot of SHs in them, this could be broken up. Okay, understand? Cysteines, they can make disulfide covalent bond. Importance of water in protein structure. You know, proteins are found inside of the cells, right? And then your cell has a lot of water molecules there. So hydrophilicity, hydrophobicity matters. Okay, the protein amino acids that have similar electropolarity, like water molecules, they'll interact with water through forming hydrogen bonds. But protein amino acids that do not have electronegativity or polarity, they do not form hydrogen bonds. We call them hydrophobic amino acid, right? So they will stay away from the water molecule. And how do they do that? By hiding inside of the protein structure. So these red balls are hydrophobic amino acid. They are normally found from inside the protein structure. Okay, and then because there are tons of water molecules out there, so egg white, and if you whip it, okay, by beating up the structure, then what happens? This structure will be disturbed, and maybe some hydrophobic amino acid will be exposed. Then what happens? Whipped egg white, so there will be a lot of air droplets could come in and interact. So air doesn't have, you know, a lot of water in there, so these hydrophobic amino acids will try to stay with the air molecule. So the whipped egg white will maintain the structure. So hydrophobic amino acid sticking with the air bubbles. Okay, that's what happens. Okay, so anyway, hydrophobic amino acids dislike water molecule. They'll try to go away from water. Understand that. But instead, hydrophilic amino acids, they don't have any problem of mingling with water molecules, so they will stay close to water molecules. That's kind of a protein folding principle, one of the most important protein folding principle. So finding hydrophilic amino acid outside, hydrophobic amino acid inside of the proteins. So we'll review about the protein structure, okay, the organization. We learned about primary structure, secondary, tertiary, and quaternary structure, okay. So how do you make those structures? First of all, primary structure is based on the chain of amino acid that are all linked together through what? Peptide bonds, the covalent bonds, right? If you put that, kind of as a primary dimensional structure, that's what primary structure is. Chain of amino acid linked through peptide bonds. Pretty simple, but protein molecules do not stay as this linkage of chains. Actually, they will fold up further. So if you look at that primary structure, we know that peptide bonds are very rigid covalent bond. Okay, so they will stay put. Carboxy and amino group linkage are very strong, but this side chain and hydrogen could rotate around on the other side. Although these two guys kind of stay put, these guys could stay around, wiggle around. So free rotation is happening on the side chain, okay, area. So that's why cysteine, if there's a cysteine there, another cysteine there, close proximity, they can make disulfide bonds. And then there's a secondary structure, of course. Not only peptide bonds, there are hydrogen bonds that can be formed. Remember those hydrophilic amino acids? If they can come together if their side chains are in close proximity, they could form hydrogen bonds easily. So the structure, protein structure based on the hydrogen bond formation is called secondary structure. You can form either a helical structure, okay, by forming hydrogen bonds in every 5 amino acid away, or four amino acid away. So every 5 amino acid, if you form hydrogen bonds, you're going to twist around your protein chain. So it exists as a helical structure. So alpha helical structure is one of the secondary structure you can form using the regular interval hydrogen bonds. Okay, so if you look at this alpha helical structure from the top, of course, you will see something like that. Okay, so there are a whole bunch of alpha helical structure in your protein complex. And then there's another structure called the beta pleated sheet structure, which is also formed through hydrogen bonds in regular interval. So if a chain of amino acid goes this way, from N-terminus to C-terminus, if there's another chain coming here, going N-terminus to C-terminus, between them, if there are hydrogen bonds formed between our side chains among the hydrophilic amino acid, yes, you can form that beta pleated sheet structure. So that's anti-parallel, meaning one protein chain goes this direction, but the other protein chain goes this direction. So they are anti, meaning orientation is different. But it could happen the parallel way, and to C terminus and to C terminus, you can still form the hydrogen bonds. So beta pleated sheet can be made anti-parallel or parallel way. Either way is fine. So three-dimensional structure of the beta pleated sheet may look like that. Okay, that's why it's called pleated sheet. Okay, so yes, you can form that. It's the secondary structure. So two examples of the secondary structure. One is alpha helix. The other is beta pleated sheet structure, anti-parallel or parallel structure. Okay, and then tertiary structure, of course, is made out of what? Beyond hydrogen bonds, there are other non-covalent bonds that can be made among the side chains, R groups of all your amino acids. Okay, those R groups can make what? Hydrophobic interactions, disulfide bonds, of course hydrogen bonds we just talked about, as well as ionic bonds. Ionic bonds can be formed between what? Acidic and basic amino acid, negatively charged and positively charged amino acid, right? Disulfide bond formed between what and what? Between two cysteines, right? Hydrophobic interaction is the interaction between hydrophobic amino acid side chains. So all these things combined, then you can form very complex structure. So not only you have this alpha helical structure, you can also form maybe disulfide bond somewhere, although it's not, marked there. There may be some ionic interaction happening somewhere. There may be some hydrophobic interaction happening somewhere. So all those things combined, you can have complex structure. Okay, that contribute to form this three-dimensional structure. So that's why I call them tertiary structure. So tertiary structure is the key for the three-dimensional structure of your protein. Okay, so again, same thing. So all these things are possible. Hydrophobic interaction between two side chains of hydrophobic amino acid, and then hydrogen bonding between the two hydrophilic amino acid, and then disulfide bridge between two cysteines, and then ionic bonds between two acidic and basic amino acid. So all those things combined, there are so many of them, you can form this three-dimensional structure. That is the tertiary structure, right? And finally, the quaternary structure is possible if your protein is huge and it's made up with multiple small polypeptides. Okay, so if your protein has subunits or multimers, meaning multiple proteins come together making a big functional protein, that's what quaternary structure is. Okay, so each subunit is actually a single polypeptide. Okay, you can split them up, and each polypeptide will give you N-terminus and C-terminus. But in order for them to maintain the complex structure and be functional, they have to come together. And how do they come together and stay together? Of course, through all those non-covalent bonds we just talked about. In the tertiary structure, hydrogen bonds, disulfide bonds, ionic bonds, hydrophobic interactions, all those things come together and play a role here in forming a quaternary structure. Okay, so quaternary structure, we were talking about the complex structure made out of the multiple subunits. Okay, so since you have multiple subunits, there may be different functions that may be done in the quaternary structure. Okay,